Excellent. All right, so today I'm going to be going over the results of my master's thesis, which is an integrative investigation into the North American Gyrinicola. Within this presentation, I got to go fix the button again. Oh, no, it worked now. We're good. Within this investigation, I had a couple of chapters to my thesis. I'm going to spend most of the time talking about chapter 1 and chapter 4. Chapter 1 will get a lot of attention because it's an introduction, which will give you a good idea of what these worms are. Chapter 2, I will cover the methods and results somewhat briefly. Chapter 3, I'll cover the methods a little bit more in depth because they're the same methods I used in chapter 4, so we don't need to talk about them twice. And then I'll give you just a little bit of the results of what we found in that investigation. And then finally, I'll spend probably the second half of this discussion talking about that final chapter. So today we are going to be going into this first chapter. Before we get started, we have to first discuss, for the people in the room who don't know, we're going to be dealing with the Oxyuroidea, which are commonly referred to as pinworms. This is a very, very diverse group of nematodes with an equally diverse group of hosts. You can find oxyurids in a wide variety of organisms, be they invertebrates like cockroaches and millipedes, or vertebrates like us humans. The Oxyuroidea you might be the most familiar with is Enterobius vermicularis, which is the human pinworm. If you have children, you've probably seen pinworm. If you were a child, you've probably seen pinworm. I'm not going to make anybody raise their hands because this is recorded, but I will say I am pretty familiar with this parasite. These pinworms get their name due to their pin-like tails. Usually the females and also the males, but they're a little bit smaller, have these very long sort of pin-like shapes to the tail. So they come to like an almost little conical point. So that's what we're going to be dealing with today, these pinworms. The specific pinworms we're going to be dealing with are the Gyrinicola. These are a globally distributed group. They are present in Asia, being found in India and Japan, Europe, with 2 species being named in France, North America, found in Canada and the United States, and South America, where there is another species which has been noted in Argentina and Brazil. All of the pinworms we'll be talking about today infect larval anurans, so you will find them inside of a tadpole. And despite this super wide range, only 6 species are currently recognized. You'll see that there's probably more than that once we get done with our results today. They are noted infecting a wide group of these larval anurans, particularly Gyrinicola batrachiensis, the North American worm, has been noted in like 18 different species of anurans here in the United States. In terms of the history of the group, the original description for the group was written in 1938, and it was described based off of Gyrinicola japonica, which was described by Yamaguchi. In that description, because it was like 100 years ago, there were some characteristics that were just not described quite accurately enough, which caused throughout the years this group of pinworms to be shifted through taxonomy many, many times. And that sounds like a little bit of complaining, but it does have implications on how people identify these worms. Because in most of the keys that we use as parasitologists, you're sort of breaking them up into like what groups they fall into. So because these worms have been shifted so much, it often leads to misidentification of these worms in tadpoles as a group as pharyngodontids or philandros, but very rarely will people actually get to Gyrinicola because some of the physical characteristics and the characteristics of the groups that they were placed in just do not match. More recently, in 2020, Saka and Nakano went back and they actually amplified some DNA from that G. japonica species and tried to look at where that worm was placed, and they re-described it. So they went back and looked at some of those characteristics. And lo and behold, they fit more with what we expect in terms of taxonomy, and they were replaced, which makes life a little bit easier for people. The specific pinworm that we'll be dealing with today is the North American tadpole pinworm, Gyrinicola batrachiensis. This pinworm is noted to be an intestinal mutualist of larval anurans. There was a study that showed that tadpoles who were infected with this pinworm emerged on average like 2 weeks earlier than their non-infected counterparts, but still had a statistically similar body size, which for the people who know about tadpoles in the room is weird. Usually when tadpoles emerge very rapidly, they have like sort of stunted arms and a smaller body form, and that usually leaves them more apt to predation. So there's some of the reason that they think this worm might be a mutualist. It helps these tadpoles who are emerging rapidly. The worm itself is a noted generalist, as I said before, having been described from 17 species of anurans throughout 4 major families of anura here in the United States. The worm itself has a really interesting reproductive mode, which has some implications for how these populations are diverging. There is going to be 2 sort of populations that's set up. In the first set of populations, there are going to be haplodiploid individuals. The females will be diploid and the males will be haploid. You usually see these kinds of populations in tadpoles that have a long time to metamorphosis. Something like a green frog or a bullfrog, which can remain a tadpole for 1 to 2 years. Within these tadpoles, the females will produce 2 different kinds of eggs, 1 kind of egg in each uterine branch. So in one branch, they will produce these thin-shelled eggs, which you can see up here. These thin-shelled eggs are retained all the way until the state in which an autoinfective larvae is formed. So they hold on to these eggs in that branch for a very long time. Second kind of eggs are these thick-shelled eggs. These thick-shelled eggs down here are usually pushed out of the female at like the 2 to 4 cell stage. These eggs are going to be what actually get passed out to the environment as a means of infecting other tadpoles. Adamson did a study and he showed that these autoinfective larvae don't survive longer than an hour outside of a tadpole. So once they've hatched, they sort of need to be in that host. Some of the cycling in these populations is associated with the seasons. So it was noted that the females usually started making these thin-shelled eggs later in the season, and then they would retain them over winter, and then in spring, they would let loose all the thin-shelled eggs they'd made in order to produce a bunch of new infection in the tadpole that they were already in. Those females produced from those thin-shelled eggs would then create more thick-shelled eggs. So it's sort of like a trade-off between what season you're depositing eggs in, and it was more likely that the females that were products of thin-shelled eggs produced thick-shelled eggs. The second group is weird. So these parthenogenic populations are going to be found in tadpoles that are really rapidly developing, something like a toad, which can go from being a little egg to a toadlet in like a month. They're very quick if they need to be. In these instances, these females don't require a male. They will do apomixis and they will produce thick-shelled eggs. However, they will only produce thick-shelled eggs in 1 uterine horn, and those eggs will then be passed out. It's sort of like a trade-off because when these tadpoles emerge, these worms actually get purged from the digestive tract because the digestive system is very different between a tadpole and an adult frog. So they have to make eggs before they're pushed out of their host. Gyrinicola batrachiensis, what we recognize as Gyrinicola batrachiensis today is a synonymy of 2 worms, Pharyngodon batrachiensis, which was first described by A.C. Walton in 1929 from Ranapipians tadpoles in Douglas Lake, Michigan. The second worm, which is grouped in with G. batrachiensis, is Pharyngodon armatus, which was also described by Walton about 4 years later from some tadpoles he was given by a colleague at the University of Virginia. Looking at them in the 80s, Adamson thought that these 2 worms were probably the same because he had just like the morphology to go off of, and he noted the presence of those thin-shelled eggs, which had not been noted in the original description. So he said, I think these are the same. I'm going to call them a synonym of one another, say they are Gyrinicola batrachiensis after these worms Yamaguchi made. Now that we know a little bit about these worms, we should talk about where they can be found, which is something I was sort of interested in because when I started this, I was told that these worms would be everywhere. And so what I did was I worked with Dr. Jason Brown to do some max and species distribution modeling, and I had 2 objectives for this chapter. The first objective was determine the habitat suitability. So what was sort of making different places more susceptible for Gyrinicola infections, and also determining what factors might influence that suitability. To do that, I took primary literature sources and also sources from GBIF, and I collected all these location points for Gyrinicola that had been either deposited or written about and not deposited, and stuff from my own sampling localities. After which I took the another set of data, which was these climatic variables, because these are commonly used to determine what is going to affect suitability for organisms. To do this, I took all of the 19 standard variables on the World Climb website at 30 arcseconds and just put them all into the ArcGIS as like separate layers and save those for my modeling efforts. I also created another model, which I thought might be somewhat more biologically relevant, which was a host layer. To do this, I took a bunch of GBIF occurrence data for bullfrogs, and then ran a very simple maxent model for it on the maxent GUI, which is the default settings, to create some idea of what the bullfrog distribution looked like in the US. I selected bullfrogs for their sort of nature, I guess. Bullfrogs are very generalist, and they are the most widely distributed frog we have here in North America. So if I was going to select one frog, and I know Gyrinicola can be in bullfrogs, I went with the bullfrogs because they're everywhere. So it might help in some of that modeling. A good stand-in for the rest of the frogs. I then ran everything using XDM Toolbox in maxent. I ran it with a bunch of different regularization multipliers to help fit the model. I rarefied all my occurrence points at 5 kilometers, so no one sampling location took over the data. And then I also did a bias layer to restrict where the model was selecting pseudo absences from. After that, I produced these two models. Yep, there we go. The first model was a model that I constructed with that Gyrinicola occurrence data I showed you and the climate layers. So for this, I ran a model using climate and Gyrinicola locations. This model is relatively good in that the AUC is somewhat high and the OER is relatively low. Normally you want an AUC somewhere around 1, but this is a wide distribution, so normally the total is a little bit lower. In terms of what I was looking for in this model, I wanted to see what environmental factors were affecting these Gyrinicola. So I looked at some of the 2 highest layers, and it seemed like precipitation and mean temperature had some effect on where these worms were actually capable of setting up infection. After that, I ran a second model in which I included the Gyrinicola locations, the climate data, and then also that model layer I had created for bullfrogs, and I let the modeling program decide what was important. I put that in, and the only thing that the model cared about in constructing where the Gyrinicola were the bullfrogs. So it no longer cared about precipitation or temperature, it just cared about that host occurrence, which was sort of interesting. There's a couple of reasons I think this happened. It is possible that there are some environmental factors affecting these Gyrinicola distributions, such as temperature and precipitation. But I'm not quite sure about that. The only time at which those environmental factors would really directly affect the Gyrinicola is in this egg stage, particularly those thick-shelled eggs. I'm not as confident in that because these thick-shelled eggs in other species have been shown to be very, very resilient. Some species of mouse infecting pinworms have been shown to alter the lipid structure of their egg layers in order to reduce water loss as precipitation goes down. And they've also been shown to be very resilient to temperatures and to withstand 2 months outside of a host before and still being viable eggs. What I think happened is that that host distribution is going to be really important, particularly because these Gyrinicola are somewhat shielded from those environmental factors, but also it might be that that host distribution is taking into account the temperature and precipitation, as well as a unique biological aspect that is that frog presence. So that's kind of what I found in that chapter. in the next chapter, I'm going to check this button real quick. All right, let's see if this works. Yeah, okay, we're good. In the next chapter, I wanted to actually get into some of the genetic data with the worms I had sampled and had those localities for. In this one, I'm going to cover the methods a little bit more in depth, and then just show you one of the trees we produced and talk about some of the results from that as well. We're working on getting this published, so hopefully you can read all of the stuff about it soon. In this chapter, I wanted to first determine the validity of Gyrinicola, so see if what we were calling Gyrinicola here in the US and Japan were actually the same genus of worms, because no one ever had shown using any form of genetic data that these worms were the same. They were very similar, sort of in the morphology, but the reproductive structures of that Japanese specimen are way different than what we have here in the United States. So there was some question about, like, are these worms really the same genus? What's going on here? So we wanted to check that. And then I also wanted to look at the phylogenetic placement to see if the addition of these North American worms would help, like, improve any determination, because in that Saka Nakano paper, they just had the one Japanese specimen that they were using to imply the location of the family. To do that, I went out to beautiful ponds like the one you see here and big boots with a big net and dug around for tadpoles. Usually Dr. Jimenez was there filming me in some manner. So he's the one who took that picture you see there. Afterwards, I would take the tadpoles back and humanely euthanize them according to our IRCOOK protocols, and then dissect them. I removed the whole digestive tract and then picked through them. Tadpoles are very easy. You just sort of like get a probe on one end and probe on the other end and pull apart, kind of like a string cheese. It makes the digestive system very open to you. Hopefully, when I went through those digestive systems, I would find a beautiful worm like the one you see here. It may not be beautiful to you, but it is to me, because that represented a lot of work of me trouncing around in the mud. With those worms, I then used a specialized extraction protocol that used a QIAGEN QAMP DNA mini kit with some modifications, because we were working with very small amounts of DNA at the recommendation from a procedure we got from a friend and colleague, Casey Bell. In this procedure, we were capable of just chopping the nematode into thirds. We saved the anterior end and the posterior end because those contain most of the diagnostic characteristics that people use to identify these worms, and then just digested the middle. By doing that, we were able to save vouchers for other people to back check our work, to use for various other studies, anything they wanted to do with them. And I was able to deposit some aspect of what I had done into museums, which is important for future researchers. Any extractions that were successful after quantification, I amplified with the following primers for 28S and 18S, which are the large ribosomal subunit and small ribosomal subunit respectively. All of our sequencing was done commercially. So this was just all commercial Sanger sequences. And then I would produce all of the contigs in sequencer. My alignments for this were done using the Kinsey protocol in Cypress. So I wanted to use some secondary structure elements to see if that would assist with any of my alignments. After that, I did all of the tree construction on Cypress as well. I did J-model test, and it always gave me GTR because I was working with a very wide range of worms in this analysis. I built 2 sets of trees and then combined them into a single figure. The Raxamel was done, and Mr. Bayes was also done. So I could have bootstrap values and also posterior probabilities to have some clade support values. Within this analysis, I had, I believe, 89 worms, all within those clade 3 nematodes. And this is sort of what the tree ended up looking like. So this is all of the worms that we included in that analysis. Within this, my worms are these worms down here. The first thing I want to make note of is that they are very well supported. So if I zoom in on these guys a little bit here, you can see that there is full support for this group. The Gyrinicola and G. japonica and what I had here in North America seem to be related to one another, which is great because we've been calling these things Gyrinicola, so they better be Gyrinicola. And following that, there was some strangeness going on. When you look at these slowly evolving genes like 28S and 18S, what I expected going into this was that I would just have one big polytomy at the base of this. So I was expecting no clades. I was expecting just a bunch of branches out of this that all represented Gyrinicola. That's not quite what I saw. As scientists, sometimes we do what I believe Dr. Sean Megan called a squint analysis, where you look at your data and you're like, oh, there's some strange pattern going on here, but that could be somewhat biased. So to look into that a little bit further, I wanted to see, okay, what is the actual diversity between these groups? Is it similar to what we see as other species differentiation? And so I did some p distances for my group, found that there were anywhere between 0 within group to 0.9% different from one another, which is relatively small because these are slowly evolving genes. But I also looked at the other groups of the Pharyngodontidae, where these worms are supposedly the family of. Among these Spaligodons, we had 0 to 3%. So I was like, I'm sort of hesitant. Among Pharyngodontidae 2, which is going to be from Batracholandros here, all the way into these Pharyngodon and other Thalandros species, we had 0 to 3% again. But then between these specimens of Thalandros filiformis and Thalandros tinifrenensis, we had, there were actually 4 individuals here. I just collapsed it so you wouldn't have even more names to look at, anywhere between 0.3 and 0.8% difference. And even within these groups, once I started to look at it a little bit more, I found that there were things that we were recognizing as separate species that were closer together than what I was seeing amongst these Gyrinicola, which led me to the investigation of the next chapter. But for this analysis, I wanted to more focus on the placement of these worms. And looking at it, we had a really hard time trying to say that this should be the family Gyrinicolidae. There was a recent publication of some new taxonomy of these worms that took a lot of work from some people, and they put them within this Pharyngodontidae group. But when you look at this, these Pharyngodontidae appear to be like paraphyletic. There's a couple of reasons that might be, and part of the reason we didn't want to say Gyrinicoidae is because there are many more Pharyngodontids that aren't represented by data on GenBank, particularly ones from things like birds and mammals and other invertebrates. So some of these deeper relationships within the group might be very unclear. For that reason, I think that they should probably be resurrected as that subfamily because it recognizes the unique grouping. It notes that these worms do have some special association with tadpoles, this strange reproductive mode. And then also, I wanted to look further into these worms because there seemed to be something going on in what we were calling Gyrinicola. There appeared to be more diversity than we would expect. So that led me into this final chapter, which is going to be on the genetic and trait variability of these worms. So in this chapter, I wanted to determine the genetic variability, adding additional nuclear ribosomal loci, as well as mitochondrial data. And then I wanted to see if what we figured out with that mitochondrial and nuclear data was supported by any morphological aspects that were overlooked before because there was no clear DNA evidence for any of these groupings. To do that, I did the same exact thing, went out to more ponds. I'm not going to go through this again, but yes, ponds. I then amplified the same 28S and 18S regions as before, as well as this new chunk of ITS1, 5.8S, and ITS2. And then I did Illumina shotgun sequencing to get a bunch of mitochondrial data. I did the ITS regions because they should be a little bit more rapidly evolving than the 18S and 28S, as they are spacers. So they can have a little bit more leniency in their mutation rate. And then I did shotgun sequencing because nothing I tried to do with PCR and mitogenomes worked at all. I tried oxyurid-specific primers, I tried nematode-specific primers, I tried a variety of things and they just failed. But we had a chance to do this next generation sequencing. So it went fine. So to talk a little bit about what I did with the next gen sequencing, I had a lot of assistance with this from Dr. Newbig, to whom I am eternally grateful. The way that this worked, the best way to go about this when you have something like these Gyrinicola, which had no really close representative available, is to work really hard to assemble like an example sequence of one of your worms. So to do that, I cast a really wide net and looked at all of that data and saw what would fit to one of the seeds. And that seed was the 28S Sanger data that I already had produced. So I used that 28S Sanger data, and then I mapped to reference all of the reads I got from the Illumina at a very low sensitivity settings, just to see what came out as possibly nuclear ribosomal. And then I took those used reads and then assembled them at higher sensitivity settings to sort of get rid of some of the chaff and just keep the good stuff in that assembly. I then used that exemplar sequence to assemble the rest of my Gyrinicola worms, so I didn't have to do the whole thing with 25 iterations over and over and over again. This just made my life so much easier. And with that example, we got this huge chunk of the nuclear ribosomal cistron. I did the same thing with the mitochondrial data, but because I did not have a Gyrinicola seed to work with, I had to work with something that was a little bit more distant. So I used the previously published mitochondrial data for Cyphacia ovulata, which is that mouse pinworm I was talking about before. And then I mapped to reference with that Cyphacia ovulata, used reads again, made my exemplar, took the exemplar, assembled the remaining Gyrinicola. I also assembled a Spaligodon specimen because in that previous analysis, it was thought that these nematodes infecting lizards were sister to the Gyrinicola, and Dr. Jimenez happened to have in his collection a Spaligodon specimen from a fence lizard in Mexico. So I got to play with one of those as well. After that, I constructed all my trees in pretty much the same manner: J-model test, RAXML, Mr. Bayes. I forgot to mention before that I did check for chain convergence. I knew Dr. Anderson was going to ask about that. I did do it, I promise. Following that, I made a variety of trees. In this analysis, I restricted the number of worms just to increase the speed, and also because we already had some sort of idea of where these worms placed. So I reduced the outgroups and was more concerned with the North American Gyrinicola. Within this analysis, there we go. These are all my worms. We did see, again, 3 sort of groupings with pretty high support. This last one here had somewhat less in the maximum likelihood support, but the other 2 had near maximum support. So it seemed like maybe we have 3 groups of these Gyrinicola inclusive with that ITS data as well. Following that, I ran a tree for the mitochondrial DNA. Lo and behold, same sort of situation where we have these 3 groups forming, each with maximum support in my maximum likelihood and Bayesian analysis. I then combined it, and again, very high support for 3 separate groupings. In this instance, this is both nuclear ribosomal and mitochondrial DNA concatenated. I did have to use 2 separate alignment types because mitochondrial DNA does not have a secondary structure. So I used a Lindsay alignment for that one, but that ended up being okay. Following this, I wanted to do something with this tree and figure out, okay, can I use this to figure out what species we might have? Is because I can look at that and think there's probably 3 species there, but if I can use delimitation analysis, which is what I did, I can figure out what is really going on between some of these patterns. So I used a BPTP analysis, which is a Bayesian implementation of the Poisson tree processes that uses the branch lengths and the rates of mutation along those branches to estimate possible species. The first thing I looked for was that it was capable of recognizing that my 3 outgroup species were separate species. If it had those together, something was probably wrong in my tree, but it did successfully do that. So I moved on to looking at my Gyrinicola. Within this, I was sort of curious because it split these 2 like it had done before, but it split that 3rd group into 2 separate species. These 2 species did have much lower support. So like with just this analysis, I probably wouldn't be confident in saying like these 2 are separate things because you have an expected value of like around 1. So 0.60 and 0.59 is relatively low. But I could use this species tree to sort of guide me in my morphological analysis, which is what I did. So the next thing I went through and did was, I think the batteries in this might be dying. The next thing I did was a morphological analysis. In doing this, one of the reasons was that nematodes are known to vary very widely between hosts, between host age. You can have the same species of nematode that's infecting different hosts, and it can look entirely different, which has led to some issues in some recent publications that have started to use some of this genetic data to infer about how these worms are related. To do this, I use several of the standard measurements that are commonly used by parasitologists to infer these characteristics of the worms. I use the tail length, so if you measure from here to the anus, you've got your tail length. The esophagus length, I use the total esophagus length in this instance. So from the end here down to the bottom of the bulb, I use the width of the pharyngeal bulb. These are going to be your feeding structures, the nerve ring, so the brain of the nematode. The excretory pore placement, you can't see it here, but I promise it's about right there. You can see it more when I zoomed in. And then the placement of the sexual characteristics, like where the vulva was on the body and how long the spicule was, as well as the max width. Because I knew that these worms could vary with host age and that the size could vary quite a bit, I wanted to standardize my measurements. So I took all of my measurements and then divided them by the total length of the individual. So a lot of what we'll be looking at is more of a proportion. So rather than saying the vulva is this long in a worm that's 4 millimeters, it's 1.5 down, compared to a worm that's 400 micrometers, it's 200 down. I divided it to get like a proportion, so it's halfway down. In doing that, I had multiple variables that I had measured as well as multiple groups, so I had to figure out what statistics I was going to use. Dr. Reeve helped me with this. I've embarrassed him. Dr. Reeve helped me with this. Very grateful to him as well. I went with a MANOVA because it's capable of differentiating between multiple groups with multiple dependent variables, and it's going to compare the variance within the groups to the variance between the groups. I went with a MANOVA rather than doing multiple ANOVAs, because when you do ANOVA after ANOVA after ANOVA, you run a higher risk of getting one of these type 1 errors when you say, this is different, but it's really just different because you just kept picking away at it. This runs just like a single statistical test and tells you, is something different between what you're calling these groups. For the groupings, my independent variable was that BPTP species identity. So those species 1, 2, 3, and 4 within that previous tree. And then my dependent variables were all of the measurements that I took of the worms. I did 7 features for 45 different female worms, which is a lot of work. It took several hours sometimes to find these little excretory pores, especially when I was first starting. You get better at it as you go on. Don't let that scare you off of nematodes. And then I took that data and ran the MANOVA on it and got a very highly significant result. What that indicates is that between what was assigned as those BTP species identities, there was some morphological characteristic that was different amongst those worms. Interpreting that is why people don't like using MANOVAs, I think, because there's a couple ways you can do it. The best way, which I was taught by Dr. Reeve, is the standardized canonical coefficients, which help you sort of break up what is contributing the most to the variance between those worms. In the first instance, most of the variance was a contrast between the placement of the vulva, the placement of the excretory pore, and the placement and, well, how wide the worm was in comparison to its total length. So it seemed like that played some influence in telling the species apart. And the second canonical vector, I like this one a little bit more because it was how wide is the bulb and how long is the esophagus. The reason I like that one a little bit more is because you can like sort of biologically interpret it. If you have a super long esophagus, you need a bigger bulb to pump the food through it, so on and so forth. So I liked that one a little bit more, which doesn't really matter. I also used these canonical coefficients plots to visualize some of this data. These are not the standardized canonical vectors, but just the regular canonical vectors. Within this analysis, you want to look for, is there differentiation between your centroids? And I did see clear distinctions between my separate groupings. So you had your species 1 in red here, which were going to be worms from Michigan and New York. Your species 2, which were these other Charleston worms and the worm I had gotten from Virginia. Species 3, which is a variety from Illinois, and then species 4, which were some worms from Urbana. I also did male worms, because male worms are what are focused more on in terms of differentiating species apart from one another in the nematode world, because they maintain most of the hard, firm characteristics. Females vary a lot and don't have anything like a spicule, which is sort of set in shape that you can pick apart. So people don't often like using them to differentiate between separate species. Within this analysis, I also had highly significant results indicating, again, that there is some difference between the males amongst these different groups. Most of the variance was due to the esophagus length and the maximum width, as well as the bulb width and maximum width against the perspective of the spicule against the total body length. In terms of what the centroids look for that, we again saw a clear distinction between these males. In this instance, it was a little bit better because they weren't even touching at all. But I was sort of excited about that. It seemed like my morphology was agreeing with what I had gotten from that genetic data, and that there was clear distinction amongst these 4 groups of worms that was probably just overlooked because there was no genetic way to interpret them before. All of my evidence is suggesting that there are more species of Gyrinicola present in the United States than currently recognized. That's going to be both the morphology and the genetics, which are in agreement. I think I have 4 likely species in this analysis, one of which is going to be still that Giba tracheensis, described originally from Michigan, one of which is probably G. armatus, which was that Pharyngodon armatus described from Virginia, because I got worms from Virginia and ran DNA for them as well. The other 2, I got to assign some names to, and we're working on getting this published as well. So in terms of the future directions for anybody else in the room or online who want to work with nematodes, if you want to work with the Gyrinicola, there's a lot of background study that can be done with what is now distinguished between these species, as they might have different effects on their host than what was previously published. So just if you take a worm from Florida and a worm from Michigan, they might not necessarily do the same thing to the frog because they are a separate species. It's common in parasites for things within the same genus that cause different effects on the host. So if you think of something like the Plasmodium, depending on what species you get, the outcomes are much different. Could be that too. You could also look at some of the reproductive characteristics over the course of the years and do all sorts of stuff like Adamson did way back in the 80s. Finally, I also recommend anybody who wants to find more parasites, you should get me some pharyngodonids from birds and mammals, so we can really work on that trait and see if we can clarify that relationship between the Gyrinicola or Gyrinicolinae and the rest of the pharyngodonids. I would like to thank the NSF Superb Scholarship for funding my research, the Southern Illinois University School of Biological Sciences. I would especially like to thank my colleagues, Dr. Newbig and Dr. Reeve. These are 2 people who were not on my committee that committed a lot of their individual time to assist me with my research, and I really appreciate that. I would finally like to thank my friends, family, especially my mother, who raised me pretty much by herself. So thanks, Mom. I think I saw you on the Zoom. If you have any questions, feel free to ask. I am here. You have access to me. I'm